Hello, virtual audience, and happy Monday. Thank you for joining us here on this lovely screen tonight. My name is Kate Bruns, and on behalf of Harvard Bookstore, the Harvard University Division of Science, and the Cabot Science Library, I am very excited to welcome you to our program tonight with Neil Shubin presenting his latest book, Some Assembly Required, Decoding 4 Billion Years of Life from Ancient Fossils to DNA. And I even have the book as a prop right here. Tonight's event is the first virtual installment in our Harvard Science Book Talk series. We are so excited to continue the work of bringing the authors of recently published science-related literature to our community during these unprecedented times. Coming up next month on May 21st, we are going to welcome Mario Livio virtually for a discussion of his new book, Galileo and the Science Deniers. And just like always, you can find announcements about upcoming events in this series at harvard.com/events/science. You can also sign up for our email newsletter at harvard.com for more updates. And additionally, we have a new Science Research Public Lecture Series YouTube page where you can see previous talks that you might have missed. This evening's event is going to conclude with some time for your questions. If you would like to ask the author something, you can go to the Ask a Question button at the bottom of the screen where you can submit your question, and Neil is going to get through as many as time allows for this evening. At the bottom of the screen during the presentation, you're also going to see a link to purchase your copy of Some Assembly Required through Harvard Bookstore's partners at Bookshop. All sales through that link support Harvard Bookstore, so a huge thank you in advance for your support during this difficult, uncertain time. Your purchases make this new virtual author series possible, and really now more than ever, they ensure the future of a landmark independent bookstore. And as always, I have a few thank yous to say. Thank you to our partners at Harvard University who really do make this series possible. And thank you to all of you for tuning in and for showing up for authors, for publishing, for indie book selling, and especially right now for science, because it really does matter. And now I'm very pleased to introduce our speaker tonight. Called a natural storyteller and a gifted scientific communicator by the Wall Street Journal, paleontologist and Harvard alum Neil Shubin has conducted landmark research on the evolutionary origin of anatomical features of animals. He's currently the Robert R. Bensley Professor of Organismal Biology and Anatomy at the University of Chicago, and he's a best-selling science author. His previous books include The Universe Within and Your Inner Fish, a journey into the 3.5 billion year history of the human body, which was named the best book of the year by the National Academy of Sciences in 2009. Tonight, he is presenting his new book, Some Assembly Required, which science calls an intimate, exciting, and thoughtful sweeping evolutionary history. Of the book, BBC Wildlife writes, "It's light of touch, anecdote-rich, funny, and satisfyingly informative. Fossils, DNA, scientists with a penchant for suits of armor. What's not to love?" We are so delighted to host this event here tonight. So without further ado, I'm going to turn things over to Neil.
Well, thank you so much. It's a delight to be here with you virtually this evening. I hope this finds you and yours well at this unprecedented time. So thank you for joining me at this time to talk about Some Assembly Required. When I was thinking about Some Assembly Required, I was, basically the idea of the whole conceit of the book is to think about the great transitions in evolution. How do we know them? How did they happen? Because when you think about, how fish evolved to walk, how birds evolved to fly, our existence on this planet, when you think about them at one level, they may seem impossible to many people. But when we break them down, we begin to use fossils and DNA and embryos and pull together many lines of evidence to see how the world came to be, in particular, how many of these great transformations actually happened. And as I was writing the book, I talk about contingency and evolution, contingency in my own life. I was reading an autobiography of Lillian Hellman. Lillian Hellman, as you can see here, had a hard living life. She was blacklisted by the House Un-American Activities Committee. She's famously hard-living human being who, in looking at her own life, had a quote that struck me as absolutely perfect for this book. And her quote is, when thinking about her own life, she said, "Nothing, of course, ever begins when you think it does." And that, in a nutshell, is how we think about evolution. Nothing ever begins when you think it does. Let me give you an example. If you think that lungs evolved to help animals live on land, at the transition from fish to land living animal. If you think feathers evolved to help animals, to help creatures, birds fly, you'd be in really good company in thinking that. You'd also be entirely wrong. Furthermore, we've known some of this for over a century, since the work of Darwin, in fact, in his sixth edition to The Origin of Species. It's a truly remarkable thing. And when we start to think about it, evolution couldn't happen in any other way. So what I'd like to do is tonight, just for the next 20, 25 minutes, and then we can answer your questions, to go through what we think about DNA, what we think about fossils, what we think about embryos, and how that tells us how great transitions have happened, and how surprising that can often be. Let's just step back and think about DNA for a second. Every single one of the cells in our bodies has DNA inside, obviously. We have trillions of cells. Let's just say, take a number. Let's say it's 4 trillion. Give or take 2 trillion. But in each of those cells, inside the nucleus of the cell, is a 6-foot-long strand of DNA that is crumbled and folded in on itself to be inside a small cell. Think about that, 6 feet. If you were to take all the DNA of our bodies, of every single cell, each one 6 feet long, from trillions of cells, and lay them end to end, our DNA individually, mine, yours, everybody's, would go essentially from Earth almost to Pluto. That's an enormous amount of genetic material inside our bodies. Furthermore, the DNA is very active. It's very dynamic. That's what we've been learning in the last decade or so. That crumpled ball, that 6-foot-long ball, is crumpled up inside the nucleus, but it's opening, it's closing, it's folding in on itself. It's actually dynamic as genes are being turned on and off as we live our daily lives. Lots of surprises there. We've lived through genome projects, which are telling us surprises about our own DNA. And let me start with one huge surprise. It's actually very relevant. Viruses on everybody's mind. And I want to open with one very surprising example. Jason Sheppard is a neurobiologist at the University of Utah. Now, Jason's not a virologist. He's not a microbiologist. Jason is a neuroscientist, biochemist, physiologist. And he's been focusing for most of his career on a gene involved in making memories. It's called ARC, A-R-C. So he's been studying ARC, and it's a very wise choice for a memory gene. Mice have ARC, and mutations in ARC, the gene, when mice have that mutation, they can solve a maze, but they can't remember the solution the next day. Humans that have mutations in ARC also have cognitive deficits and other things, suggesting that's also involved in our memories. And it's seen in other animals as well. So Jason was looking at ARC, and he was looking at the protein that the gene makes. And so he popped the protein that the gene makes, he isolated it, popped it on a slide, and he was kind enough to send me the slide that got him going on this. This is the slide ARC. He popped it under the microscope, and you can see there are little clumps all through the slide. You could see those circular clumps. He was looking at those circular clumps, and he was thinking, seen those before. So he dug out a textbook, and lo and behold, he had seen them before. Here's a slide. He sent this one to me as well. You see the arrow pointing to clumps. These are virtually identical to the clumps made by the ARC protein. But the surprise here is those clumps are made by HIV, the virus that causes AIDS. He was surprised, obviously. So, he's at a medical school at the University of Utah. He calls colleagues at a building next door who are in virology, actually experts in HIV biology. He invites them over, shows them his ARC slide, doesn't tell them what's on it, says, what do you think that is? They say, oh, that's HIV, virus that causes AIDS. He's like, nope, it is a memory gene in mice, and you have it too. So they studied it more. They studied the structure of the protein. They studied the structure of the gene. And it turns out that ARC is a repurposed virus. One of our genes, the genes that's involved in memories, was once a virus that when they looked at, mapped its structure, and they looked at its distribution in other animals, the hypothesis is, supported by lots of data, that at some point in the distant past, a virus invaded the genome, went into the genome, but was later, instead of being infectious, was later repurposed. How, by some means we don't know, was repurposed to function in memory. So a gene involved in memory was once a virus that invaded the genome. Now, that's not just one surprise. There are other surprises as well. If you look at proteins involved in the placenta during reproduction, some of those were ancient repurposed viruses as well. We see this again and again. And there are so many surprises in our genome. Turns out if you look at our genome, only 2% of our genome contains genes, the genes that code for proteins. There's a whole 98% of stuff that does other things. Very important, but it does other things. Well, when we look at that, Turns out that 8% of our genome were ancient viruses that invaded the genome, invaded and then got knocked out, no longer infectious, but they sit there like fossils in a graveyard inside the genome. That is, we have 4 times more genetic material derived from viruses in our own genome than we do our own genes. And that's just the tip of the iceberg of surprises. These are the sorts of surprises that we see. Nothing ever begins when you think it does. And that's so genes involved in memory were once ancient viruses. But there are other surprises as well. And these relate to one of the great mysteries of biology, which we've made incredible progress in the last several decades thinking about. You know, for us, it all begins here, right? In a fertilized egg, a single cell. That single cell, through the course of embryonic development, eventually gives rise to an individual with trillions of cells, and each of those cells packed in the right place. There are brain cells in the right place, nerve cells in the right place, our eyes, muscles, and so forth. So it's this highly organized collection of an enormous number, trillions of cells. We call this process of going from one cell to a 4 trillion cell body, we call that bodybuilding. So to go from the thing on the left to the creature on the right, a lot has to happen. And much of that depends on the DNA inside the egg, because DNA inside the egg is turned on and off during the process of development. Genes are activated, genes are turned off, genes interact with one another. And it's those interactions which produce the tissues and diverse repertoire of cells inside our bodies. The process of development has been so important to evolutionary biologists, you know, in the last century and a half, as we think about it. In fact, we now have tools that can probe that at the molecular level. And let's just work at it from a classical level, as some of our predecessors did over a century ago, and think about it at the level of molecular biology. Now, there are many classical biologists who thought about development as it relates to diversity. But one of the great way stations here is really with the work of a herpetologist, Auguste Dumares. He was a keeper of reptiles in the Museum of Natural History in Paris. And he was very fortunate to be in Paris at this particular time in the 1800s, right around the Darwinian time, about 1859, 1860, and so forth. He was recipient of many organisms that expeditions were collecting, and they'd bring them back to Paris, and he would study them and rear them. Well, one day he got a shipment of salamanders, such as this one here. These were collected from Mexico. And the reason why this shipment of salamanders was given to him was because the researchers saw that this salamander is an adult salamander, a big one. It can sexually reproduce, but look at it. It has external gills. It has a big fleshy tail, clearly aquatic. So they gave them to Dumares thinking, well, you know, this is a sort of a half aquatic, half terrestrial creature. You know, maybe this can inform Darwin's new theory of evolution. Because by studying it, maybe you can understand how fish evolved to walk on land. Great idea. So, you know, Dumares kept the salamanders. He put them in a menagerie, took care of them. He came back at some time a little while later, and he found, to his great surprise, not just one kind of salamander in the enclosure, but there were 2 kinds of salamander. There was a new fully mature, sexually mature adult. This one was fully terrestrial. This one had no external gills. This one had a totally different tail. In fact, the whole body looked different. The mouth and feeding structures looked different. Whole thing. 2 different kinds. It's almost as if someone put a chimpanzee in a cage, you know, 1 month, and then came back a little while later and found chimpanzees and gorillas cohabiting the cage. Really remarkable. So Dumares was a good scientist, and he thought about, well, how could this possibly happen? Well, of course, he looked at development, and he saw something very important. When he looked at development, what he saw is that, you know, salamanders begin in eggs. They hatch as larvae, as you can see here. Those larvae are aquatic. And you can see those larvae have, you know, external gills and big fleshy tail. Well, normally development goes on, and then the salamanders metamorphose. They metamorphose, those little tadpole-like larvae, metamorphose to a terrestrial adult you see on the left. But if you don't metamorphose, then what you have is a big, sexually mature and aquatic adult. So it's a simple shift whether the animal metamorphoses or not. What's happened inside that enclosure was a simple trigger of change in a level of a hormone that basically changed whether the animal metamorphosed or whether it didn't. The big thing you see here is a simple change to development can have massive ramifications across the entire body of the animal. And this kind of study really set off in a whole new area of research. People in the field really looked at how subtle changes in the timing of developmental events, maybe stopping early or extending, looking at subtle shifts of development, how they can have profound impacts on evolution, on the whole body itself. Well, now we study development using a variety of molecular tools. And what I wanted to do is to talk a little bit about what those molecular tools are showing us about how development can evolve. So we can look at genes, and throughout the genome, we can characterize the entire genome. We can now look at individual tissues and see all the genes that are turned on in individual cells and whole tissues. We can begin to map what happens to DNA as it turns on and off, as genes are turned on and off, as the genome opens and closes. And as this slide suggests pretty graphically, is we can now edit the genome. We can knock out genes, we can rewrite the genetic code in many different ways. Not with tweezers, we do it with enzymes and little pieces of RNA, but the result is the same. We can cut and paste different parts of the genome. So what does this tell us about development? Well, I'm going to talk about some studies that were done by a colleague of mine, Nipam Patel, at the Marine Biological Laboratory in Woods Hole, Massachusetts. So Nipam is interested in development and evolution. And he's, for part of his work, has focused on this tiny little creature. It's about 1 centimeter long. Sorry, I don't have a scale on it. And it's called Parahialli. And look at it. You can see its head is on the left, its tail's on the right. These are creatures, if you've seen them they're called amphipods. If you've seen them like dig in the sand in Cape Cod or whatever, you'll see these little clear things that jump around. My kids used to call them jumpies because they jump around. The tiny little things jump around. But what's interesting is, look at that. They have all kinds of different legs. They have some forward-facing legs, some backward-facing legs, some legs that kind of look feathery. And he really wanted to see what is controlling the legs in Parahialli. Can we make any inferences from that? What he found was that, and that's why I want to focus on this a little bit, is that there is a genetic address for the different legs in Parahialli. Look at the red arrow. The red arrow is pointing to backward-facing legs. And that is an area that's been coded gray because a gene, UBX, which you can see it's labeled here, is turned on in that area. So where UBX is turned on, you get a backward-facing leg. In the area where there's a blue arrow, there's forward-facing legs. And that is an area, you could see it's hatched there, where two genes are turned on, UBX and ABDA. That is, when you have those two genes in that genetic address, you get a forward-facing leg. And then there's legs in the back. What Nipam did was he used those gene editing tools to change the genes that are active in different parts of the body. And what he found was he can control which legs form in each segment. Really remarkable stuff. So that when he knocked out ABDA, he got rid of that. He basically turned that whole midsection into an area that just is gray for UBX. And he essentially made an animal that has only backward-facing legs and no forward-facing legs. So really that subtle shift in changing a genetic address in the body controls what organs form in which place. Really remarkable stuff. And it's a truly elegant study. And this has been done with other creatures as well, manipulating the genome to manipulate the ultimate form. Now, why this is particularly important with these genes is because these genes are present not only in Parahialli, but they're present in flies. And you can see we've color-coded it here to show how these genes are turned on in different parts of the body in the embryo, which you see in the top, which is that little ovoid thing. And then you can see what it looks like in the adult. But it turns out that we have versions of these genes too, along with mice and reptiles and birds and so forth. We can see them active in our embryos. And what are they doing? They're patterning our body axis, in this case, the vertebrae from the neck region, the cervical region, to the tail. So this is truly sort of a universal code in some ways. Versions of the same genes are working in flies and vertebrates, as well as in mice and people. Well, can we do the same experiments that Nipam did in mammals? Well, we won't do them in people, but they've been done in mice. Let's look at it. So here's a mouse, a cute little mouse there. And you can see we're focusing in on the vertebrae that are in the base of the back, near the base of the tail. Let's zoom in. We're going to look at genes, and we're going to look at genetic addresses to see what's happening in the vertebrae. So now we're looking at these genes. They're called Hox genes, H-O-X. They have different names. But what you're looking here with the red arrow is pointing, you can see there's a gene called Hox10, which is turned on in the back area. and a HOX11 area. So there's 3 regions that you see genetically. There's an address where only HOX10 is turned on, an address where, and further in the back, where HOX11 is turned on, and then where the arrow is pointing, there's an area of overlap where both HOX10 and HOX11 are both turned on. Well, it turns out the area that only sees the activity of HOX10, those vertebrae become what are known as lumbar vertebrae, the base of the spine. The area where we have an overlap between HOX10 and HOX11, that area becomes sacral vertebrae. Well, let's do the same experiment that happened with Nepom and Parahialli. This is done by a team at the University of Michigan. If they make a mouse that only has HOX10, that is, there's no HOX11, the prediction would be you'd have a mouse with only lumbar vertebrae, that those sacral vertebrae were turned into lumbar vertebrae, and that's exactly what happens. The sacral vertebrae were transformed into lumbar vertebrae. So basically, nothing ever begins when you think it does. The genetic elements that control the formation of our vertebral column, in fact, much of our body axis, actually had their deepest roots in creatures such as flies, and a common ancestor we share with flies and Parahialli and other things like that. Deeply, deeply ancient. So we're thinking, what does all this mean? So let's just put some of this together, and then I'll take your questions. This diagram shows kind of in a cartoon form of what we think about the transition from fish to tetrapod. In fact, this was the limbed animal. This was done in the 1980s. We now have many more fossils. But when you look at this as just the endpoints, fish on top, limbed animal on the bottom, it seems kind of impossible, right? Think about what has to happen for animals to crawl on land. You know, animals have to have lungs. They have to have arms and legs with wrists and ankles and fingers and toes. They have to have a neck. There's all kinds of structures that need to come about for animals to be able to walk on land. When you think about the long list of changes that have to happen, it almost seems impossible that that could have happened, to back in the Devonian 375 million years ago for creatures to walk on land. But there are real surprises here. Again, nothing ever begins when you think it does. Turns out if we look at fish, many fish, by the way, this is a lungfish, Australian lungfish. I want to hug it, okay? It is arguably the cutest little fish ever. These are really adorable fish. They're adorable in a lot of ways because they have gills, but they also have lungs. And it's been known for over a century that they have lungs. Their lungs are used to breathe air. They use the lungs to breathe air when the oxygen content of the water is not sufficient for them to breathe with their gills. So they have both lungs and gills, and they trade off between one and the other. You might think that that's a one-off and just have one kind of lungfish. No, we have at least 3 different kinds of lungfish. And indeed, when we look at the tree of life, or when we look at their structure of their lungs, what you can see is their lungs are bilobed like ours. They have alveoli like ours. They're structured like ours. And in fact, many of the genes that are behind the formation of their lungs are similar to the genes that are behind the formation of our own lungs. And when you look at the distribution of lungs in the record, evolutionary record, this is an evolutionary tree, and it's highly pruned. On the right, you see a limbed animal. You could be there, a lizard, anything with limbs is there. Then the lungfish are the next lower down, and then you have other kinds of fish. If you look at the distribution of lungs in fish, that's what you see. That is, lungfish have them, and also all kinds of primitive ray fin fish have them. Indeed, there's suggestion that some very ancient fish in the fossil record had lungs. So lungs were around in the evolutionary record well before animals ever took their first steps on land. They arose to help creatures live in water that likely had a variable oxygen content. And the story doesn't stop there. That is, to flesh out this diagram, my colleagues and I would go to places like this. This is the Devonian of Ellesmere Island. We led expeditions there for a number of years. We found fossil like this. This is Tiktalik Rosia. We now have 20 specimens of this. Other teams have found similar fossils in other parts of the world, namely in southern Quebec, as well as in Eastern Europe and Latvia. And when we look at these things, we see here's a fish that has a neck. You could see its fin right there. When you crack open the fin, it has bones that correspond to the upper arm, forearm, even parts of a wrist like ours. This is a fish that could walk with these appendages and even support its body. A fish with a neck, a fish with a neck, a fish with fins with arm bones inside, a fish with both lungs and gills. This is a fish that basically had all the tools needed to live on, walk on land, but it was still living in water. What does this mean? What this means is that fish living in aquatic ecosystems in the Devonian already had lungs and wrists and arm bones and necks and all the stuff needed to walk on land, such that when the opportunity came, when those when those features were needed to walk on land, all they had to do was change their function, that they were repurposed for that. Much of evolution does not consist in the origin of new structures, new inventions. It comes down to using old inventions in new ways. And evolution could not happen in any other way. And in fact, when you think about birds, I'm just going to end here with this case. Birds, what do they have? They have feathers, they have wings, they have high metabolisms, they have hollow bones, they have wishbones, lots of features. If you were to think about that, you think, well, how could those features all come about at the same time? Birds would never have arisen that way. But it turns out all those features came about in dinosaurs, very fast-running theropod carnivorous dinosaurs. They had feathers, they had wishbones, they had wing-like structures in their bodies. Many of the inventions that birds used to fly arose in dinosaurs living on land. The story is the same as the one with tetrapods. And that's why Lillian Hellman's statement is nothing, of course, ever begins when you think it does, is so prescient, because it really solves a problem for evolution. That is, much of evolution consists of repurposing, copying, modifying structures that already existed. And much of this comes down to changing functions. So you can ask the question, why should I care about any of this? Well, there's a reason why we should care, because much of human health depends on our knowledge of evolution and our connection to the rest of life on our planet. If you look at the Nobel Prizes that have gone to medicine and physiology, to the discoveries that have really affected our health and well-being, who have they gone to? They've gone to people working on mice. They've gone to people working on flies. They've gone to a person working on corn. They've gone, in fact, 2 Nobel Prizes awarded to 5 people in the last 12 years have gone to folks working on Caenorhabditis elegans, a tiny little worm the size of a comma on a piece of paper. Yet that little worm is telling us how our genes are turned off in health and disease, and what goes wrong in diseases like cancer. I like to think that as we discover cures to everything that ails us, from Alzheimer's to different cancers, that the breakthroughs that will extend and enrich our lives will in some way be based on flies, worms, and in some cases even fish. I can't imagine a more powerful or more beautiful statement on the importance of evolution than that. Thank you very much. And I'm happy to take your questions. Let's see what we got. Am I doing this?
Yes.
Awesome. Are you going to curate the questions, or should I just answer?
I can pick some and read them out to you if you'd like.
This one with the 15 votes looks pretty good. Yeah. What do you think secondary science teachers can do to make evolutionary biology more relevant and engaging for our teenage students? Sometimes this topic is a tougher sell as compared to other topics with more hands-on lab options. Good question, great question. I think the power of evolution and the resistance to understanding evolution, which you sometimes get for students, disappears or is mediated when we focus on the stories of discovery. When we focus, so for instance, when I talk to my classes, I talk about Tiktalik. In fact, I bring Tiktalik. And so I love to tell the discovery stories of how we predicted where we'd find it. Then we found it. We use the tools of evolutionary biology. I think the more you can focus on discovery stories, whether it's about genetics, whether it's about embryos, whether it's about functional anatomy, whether it's about fossils, the more you focus on that, I think you can tell the human stories that are behind discoveries. When people got lucky or worked hard, or or it's just the human stories of making mistakes and learning from failure. All these things have a resonant that appeal, and I think it's much easier to teach the evolutionary biology when we focus on how we know, right? Because how we know is as important as what we know, and I think the more we can get into that space with our students, the better we'll be. Will there be signed copies of the book on sale by any chance? Well, when I have a mailroom here, I'll be more than happy to sign your books if you mail them to me. But I unfortunately have no working mailroom here at the office. Will COVID-19 change our human DNA? Well, one thing we know is that many of the proteins and many of the cellular processes we have going on inside us happen in response to a virus. And it's usually through evolution, much of the evolution that's going on inside of us. And that there are signatures of natural selection going on inside our own genomes. We see it in our metabolic processes, we see it in physiological processes, but we also see it in the balance with microbes, with bacteria, and yes, with viruses as well. So there's a paper published a few years ago out of Stanford showing like the signature of natural selection on on some of the cellular processes that go on inside of us, and that was hypothesized to be in response to sort of viral invasions and so forth. So I have no idea what COVID-19 will do, but that would be one example of how viruses affect us. Do you think viruses should be part of the tree of life? Viruses are so Peter Medawar called them trouble wrapped in a protein. Basically, they're tiny little bits of genetic material, not much genetic material, usually wrapped in a protein. They are sort of sit at the margins of what we think about living creatures, right? They don't really do anything until they find a host cell. Then once they find that host cell, then a whole chain reaction is turned on, where they essentially turn the host cell into a factory to make more viruses, right? And they have different ways of doing it. There's a whole diversity, the huge diversity of these things. In fact, when somebody counted, this is an aside, there are more viruses in the ocean than there are stars in the known universe. That's pretty amazing. I saw that calculation, it blew my mind. But anyway, there's a lot, and they're also highly diverse. But they're little machines to turn their hosts into factories to make them themselves. But they don't do anything until they find a host cell. So it's almost like they're not alive until they find a host cell. So I wouldn't put them on the tree of life just because of that alone. But there are other reasons because I don't like them. Any idea? I don't know how long viruses have been around and how they get started. They've been around for a while, but there's two theories. Either they're really primitive, or like a lot of parasites, they're stripped down. So there are a couple different theories of that, but they've definitely been around for a while. I mean, the ARC gene, by Jason Sheppard's work, he suggested that the ARC gene invaded the genome of a distant ancestor that we share with fish 375 million years ago. But there are other signatures of viral invasions that go back even further. What are a few of the most pressing questions in the science of evolution? Well, there are a lot of great questions about evolution. I mean, one is, there are a couple really big ones. The one is how random is evolution? If you were to replay the tape of life, will we get to the same state? One thing we're seeing when we look at evolution is we see that, oftentimes similar structures evolve independently in different creatures. So evolution doesn't appear sometimes to be as random as we'd like to think. So one issue is that how random is evolution? Are there loaded dice to evolution? That's one big one. Rates of evolution is another big one. Why do some types of species, types of groups evolve very rapidly and leave many different kinds of descendants and huge branches of the tree of life? Why do others not change at all over long periods of time? That's a very big one as well. Other big questions are mass extinctions. What controls how selective extinctions are? What controls why some creatures live and why some creatures die in mass extinction, such as the one that wiped out the dinosaurs? So lots of open questions, which is good for people like me because that keeps us in business. One of the questions I really like to think about that'll probably occupy much of my future is the origin of vertebrates. How did creatures with backbones and skulls, those kinds of skeletons, how did they come about from worm-like creatures? We have some fossils, but not a lot. So that's, for me as a fossil hunter, that's like perfect because I'd like to focus on that. And the time periods probably you do it would be late Cambrian, early Ordovician, things like that, where people haven't looked. So yeah, so stay tuned to this channel once I can get out in the field again. Let's see what else we got. Yeah, so my son wants to understand why, as a paleontologist, you need to study biology as you want to know much of the biology of animals. Yeah, so paleontology, to be a paleontologist, there's really two pathways, both of them really important. You need training in both. Some people take it from the biological side, which is what I do. Others people take it from the geological side, which is also what I did, but I just didn't major in it when I was a student. But it's to be a paleontologist, you really have to be as fluid with and as facile with geology as you are with biology. They're the two, you can't really separate the two disciplines when you're working as a paleontologist. And I find that that's what's invigorating about it, right? Because it's multiple disciplines to make it work, and it's always exciting that way. Because when I'm in the field looking for fossils, I'm wearing my geology hat, right? We're trying to figure out which rocks are going to hold the fossils, and we're trying to think about what environments are represented here. Trying to figure out what places are most likely to hold the best fossils that are well preserved. But once I find those fossils, I really have to think about the evolutionary side. How did these things evolve? How do they function? What was the ecosystem like? What other creatures were they living with? Who was eating whom? That kind of thing. And so, and how did they work? How did they walk about or swim or what have you? And that's all biology. So what I love about paleontology is it's really kind of a fluid mix of biology and geology. My lab is also molecular, so we also work on Hox genes, the ones I showed you in the vertebral column. We work on those genes as well. And so really when you're a working scientist, honestly, it's about the questions. And the questions for me are, how did the big evolutionary changes happen? The tools are finding fossils, like this one here, studying DNA, studying how creatures work, how the biomechanics, the kinematics of how they might have moved about or fed or what have you. So that's what's exciting about it, is that it's just not one discipline. I'm trying to, we're getting tools from all over science to answer these fundamental questions. So for me, it's about the questions, and then I try to collect the tools, learn the tools to answer those questions. What else we got going here? Let's see. Dr. Shubin, 8, but I'm your biggest fan. Oh, thank you. I've been watching your interfish since I was 3. Oh my God. Do you know any camps for becoming a paleontologist for young people? No, I don't. It's a great idea. I know there are summer courses that are taught by museums. Like there's one here at the Field Museum called Stones and Bones, and it's for students, and they have a part classroom experience for like a week or two, and then they go out to Wyoming and work for a few weeks there. And I know there are a number of programs like that at other museums. So I'd encourage you, if this is your interest, I'd encourage you to check out museums and what sort of programs they have for summer work. Also, there are some summer schools. Will you be hosting any more Zoom chats for science teachers? Yeah, I will, most definitely. So what happened was, when my book tour was canceled, this is actually my first book talk, and my book came out March 17th. When my book tour was canceled, I realized I have all this time in my hands. I was going to be traveling in these different cities. And so I realized teachers need content. You're switching to remote learning. So basically, you can email me. You find it, just Google me. You can email me. I'm more than happy to schedule a Zoom drop-in and do a Q&A with students who are learning about Tiktalik or using my books or TV show or what have you. It's all fun. I've done about 20, I've about 70 more scheduled through May. And yeah, it feels good at this time, in the age of coronavirus, to give back in that way, and to meet students and teachers and share their enthusiasm for evolution and learning about evolution. So yeah, please get in touch with me. That'd be awesome. In fact, I got about 10 this week we're doing. Who has been the most influential scientist in your life? Good question. I've been really privileged to work with some amazing people. I think so. When I was at Harvard, I worked with a professor named Farish Jenkins. He was a paleontologist at the Museum of Comparative Zoology there. Museum of Comparative Zoology is an amazing institution, just an incredible place. I was fortunate to be a student there in the 1980s. I worked with Farish. He was the one who taught me fieldwork, like particularly extreme fieldwork, like in the Arctic. So much of what I've done, I've learned, with Farish. But there've been other people who've been influential in other ways. There's when I went to University of California after I graduated Harvard, I went to Berkeley for a period of time. I was very unfortunate to work with a scientist by the name of David Wake. David Wake is just a phenomenal scientist and a great human being. And he is just like an evolutionary biologist. He just knows the field so well. And his lens in evolutionary biology are salamanders. He knows pretty much everything you need to know about salamanders. And they're a window into a whole world of evolution. So yeah, David Wake was another really big one for me. So one paleontologist, one neontologist. Hence the salamander example, that was a reference to David. And, how does the epigenome, to the extent that we know it today, contribute to the turning on or turning off of the genes you've been talking about? Yeah, that's a good question. So much of what we're doing here is not really, the epigenome is part of that. So there are epigenomic changes that happen. But for the most part, it's other parts. It's transcription factors and other things. The lasting kinds of changes that we see are not epigenetic, but many of the changes in gene activity can relate to epigenetics. And the question how Hox genes, like these developmental genes, have been affected by epigenetic changes is really actually only being recently explored. So I don't really have an easy answer to that. You said lungs arose to help creatures live in water. What were the first lungs repurposed from? More generally, how did genuinely new traits arise? Well, there's nothing genuinely new. Okay, that's number one. Everything is repurposed something else. That is, everything has antecedents, and those antecedents, as Lillian Hellman said, kind of don't look the way you necessarily think. But let's step back a bit. Fish that don't have lungs, many of those fish that don't have lungs have another kind of air sac that lies adjacent to the gut tube, and that's a swim bladder. And it turns out the swim bladders and lungs are very, very similar developmentally. That is, if you look at the early developmental stages, both begin as out pockets of the esophagus, the gut tube. One migrates to the back, the so-called dorsal surface. The other migrates to the ventral surface. So lungs are more ventral. Swim bladders are more dorsal, but they share so much. So the primitive thing is probably generally air sacs that butt out from the gut tube. You mentioned that all those primitive arm bones existed in fish prior to the move to land. They did. What was the selective pressure of purely aquatic fish to begin developing those arms without walking on land? Well, remember what these fish are likely doing. They're likely moving about with appendages in water. That is, a lot of fish walk on the water bottom, or they're maneuvering through weed-choked streams and swamps and so forth. So they're clearly using the appendages to move about. And a form of walking actually arose in fish well before animals walk on land. What we see is alternate gaits in fish, and many different fish that walk on the water bottom. So it turns out we think that a lot of these fish are actually maneuvering on the water bottom, maybe even the shallows, and maybe even partially in the mud flats, using arm and leg bones set in a fin. So they sort of had a multi-purpose amphibious kind of organ. Let's go back. Is some assembly required written in a story-like format? Yeah, it is. So go to the Amazon page and you'll see like an excerpt of a chapter. You'll get a sense of how it's written. What I like to do is tell the stories of wonderful people. And I like to use a couple people like that to tell the larger story of science. So one story I'll just tell you, it's one of my favorite ones, which I didn't talk about in the book talk, but I just love it so much, I'm gonna give it to you now. Julia Barlow Platt. Julia Barlow Platt was born in the mid-1800s went to the University of Vermont, and she wanted to become a biologist. And she ended up going to Harvard for graduate school, but she couldn't get a PhD. She couldn't get the final degree. The reason why is they weren't offering degrees to females at the time. So undeterred, this is the story of her life. Undeterred, she went to Germany and finished a PhD there, where it was more friendly to women, and then came back to the United States and worked at the Marine Biological Laboratory. This is how I found out about her, when I was at the Marine Biological Laboratory. She went back to the Marine Biological Laboratory in the late 1800s to work with, in early 1900s, work with O.C. Whitman, who was the director there at the time. He was also a professor here at Chicago. But with Whitman, she started to develop techniques to trace cells during development. And she was looking at head development in salamanders and sharks. And she was tracing cells, and she found something surprising, that the cells that gave rise to some of the bones in the skull weren't coming from where people thought they would be. They were coming from a different place. And that doesn't sound like much, but it was huge, because people had set theories on where cells that gave rise to skeletons came from. And she was showing that they weren't coming from the known set of cells. So she published a paper like that, and it was uniformly derided. I mean, she was insulted, and to the point where, she was like begging people to stand up for her. So a famous biologist did from Anton Dorn, a very famous biologist, defended her, but it was too late. She couldn't get a job in science. She ended up writing David Starr Jordan, the president of Stanford University at the time, begging him for a job. I almost cried when I read her letter. She was literally saying, I have to leave the field. I made this great discovery, but no one believes me, and which just went on. She couldn't get a job. And so she ended up leaving science and moving to Pacific Grove, California in the early 1900s. In Pacific Grove, California, she became mayor of Pacific Grove, California. What did she do? She saved Monterey Bay. I mean, wow, it's amazing. And she was vindicated by other discoveries that showed that those cells that were giving rise to the crazy part of the brain, the crazy cells that were giving rise to the skeleton, which were surprising, were a whole new different type of germ layer called neural crest. And it was one of these stories. It's like, oh my goodness, here's a person who never gave up, who made great discoveries, and found a way to contribute. I love telling stories like that. So yeah, stories like that are in the book. So long way of answering your question, but I had to tell Julia Barlow Platt story because it's amazing. How did you begin your career in education and studies in evolution? Well, my career in evolution began when I was in college. Really, when I went to college, I took a class. I was in New York. I went to Columbia. I went to New York. And my freshman year, I took a human evolution course taught by a curator at the American Museum of Natural History. So I went to the curator after the class and I said, "Hey, my name's Neil Shubin. I'd like to be a paleontologist. Can I volunteer for you?" And he's like, "Sure." He said, "Yes." I was like, "Okay." So I went and worked in his lab, in his collection area. And then I got invited on a dig and went and collected fossils while I was a college student in Wyoming. And I was an utter disaster. I just got nothing. I destroyed more fossils than I found, but I liked it enough that I wanted to stick with it. So I decided go to graduate school. That's what brought me to Cambridge. Yeah, and then one thing led to another. That's how I, got into science. And then once I was in graduate school, thinking about paleontology, I was led to think about embryos and embryology, kind of like the story we talked about today. How to get into education? Well, one of the things is the more you study a field, the more, and if you love a field, there's nothing better than to talk about it and to teach it to students. So education naturally flowed, from a passion for evolution and paleontology and embryology and things like that. And so and so I teach courses here. I teach a course in human anatomy to undergraduates. It's for evolutionary anatomy. I teach a course here with a cosmologist about from the Big Bang to human evolution. We go through like origins of everything kind of thing. So it's a really fun course. It's in the fall. 10 weeks, 13.7 billion years. What is a misconception you often find yourself fighting against? Well, one big one is what you heard about today, that there's like these missing links in evolution, that's a continual progress of, one mutation, that the great inventions of the history of life arose at the great revolutions. That's not the case. The great inventions that have propelled the great revolutions in the history of life always came about well before the revolution that you're interested in. So, lungs aren't involved. Came out well before the invasion. Fish evolved to walk on land. Feathers well before birds ever flew. That kind of thing. And you see it in trait after trait. And by the way, that applies to genes as well, right? So the genes that are patterned much of our hand and feet, and mice and us and so forth. They arose in fish. In fact, they even had a deeper origin in flies and vertebrates and things like that. So yeah, lots of surprises. I wonder about the title "Some Assembly Required." The phrase "is just a sense of agency is involved as a do-it-yourself project." Nah, I was just choosing it because it's I enjoyed. I like the "Some Assembly Required" as a parent with the kid. I spent a lot of time trying to put together toys that never worked. Basic components provided, must combine them yourself. Yeah. So when you're talking about evolution and evolution development, you could talk about developmental processes. Think about it as a recipe, right? A recipe has processes, but it also has ingredients. So the ingredients are the proteins and the genes. The processes are the ways that those genes interact and the way that cells and tissues interact to build organs. Yeah. So. How do you handle deep time or geological time? I find that is one of the hardest concepts to understand, but one that allows for the diversity we see today and in the fossil record. Yeah, deep time. There's so many great teaching tools for deep time, and we always use analogies. So Carl Sagan always used the Earth, the year, the cosmic year, from the Big Bang to today, 13.7 years, scale it to, a calendar from, January 1st to December 31st. That's a great way to do it. I've had colleagues who do it with a roll of toilet paper. They take the roll and, somebody takes one end, they just walk across the lecture hall, and they have them stop and rip off toilet paper for different events, how much paper there is. So there are different analogies. People have clever tools to do it, using different analogies to teach it with. All of them, come out with, we humans have only been here for a sliver of the history of either life or the cosmos. What are the biggest challenges between scientific writing journals versus popular science writing books? Oh, gotcha. So this whole talk, my whole book, is based on a concept called exaptation. Some people call it pre-adaptation, which is jargon for change in much of evolution happens by changes in function of pre-existing structures, not new structures themselves. Inner Fish was a whole book about homology, how we compare similar features in different critters, right? Yet in both books, I never use those words. And that creates opportunity and that creates problems. because we use jargon in science because it's so precise. There's so much information when I use a scientific jargon word. People know exactly, they know the history, they know my meaning, they know where I'm going with it. You know, there's so much unsaid in a single word. Now, when you remove, but jargon can really hurt your science writing for a general public because people can get tripped up on words. You're slowing them down. It's a word that's kind of between them and the concept. And so I'll use jargon. I'll use the jargon judiciously when I have to, but I prefer not to. And the workarounds are really tough because once you get away from jargon, you're kind of walking on a tightrope. You're walking on a tightrope without a net. So you have to use analogies, you have to use stories, you have to use other things to tell that, you know, if you're not going to get into the jargon. So I find that's one of the biggest challenges. Another really, really big challenge is how much detail to layer in. You know, because we scientists, you know, it's the details that I love. You know, that's the details I'm studying. You know, I study, like, you know, when I look at Tiktalik, you know, I'm studying the details of individual bones and, you know, and what's in the head and how they get together. You know, that detail is important to me as a scientist. But, you know, for me to communicate Tiktalik to the general public, you know, those details, you know, where do I stop with the details? So that's one challenge oftentimes is, you know, kind of know when details are important, when they're not important, you know, and sometimes you get it right, sometimes you get it wrong. That's the best thing I can say. How did you know where to look for Tiktalik? Yeah, so we looked for places in the world that had rocks of the right age to answer the question. So basically, in late Devonian, we knew the late Devonian was the time period when fish evolved, middle to late Devonian when fish evolved to walk on land. So we looked at that age rock, about 375 million years old. We looked for places in the world that had rocks of the right age to hold the fossils. I mean, the right type to hold the fossils. So not every kind of rock holds fossils. Some are superheated, some are super squeezed. You know, some don't reflect the right environments. So we look at rocks the right age, rocks the right type. And then we go to places where the rocks are exposed to the surface, like deserts and things like that. So you apply those three filters. A big world with lots of rock becomes a lot smaller, and you can select a handful of places. Turns out the Canadian Arctic was absolutely perfect, as is the Antarctica as well, which is where we're working now. Well, not now, but where I've been working. Did Tiktalik look like you expected it to? Yeah, it did. We were looking for a flat-headed fish. And so, yeah, when I first saw Tiktalik, this one here, we were looking for a flathead. You know, so the first thing we saw in Tiktalik was this little V in a rock right there. But I saw these teeth. It's actually this specimen. This is a cast. I saw these teeth and I saw the snout here. And I knew when I saw the snout that it was a snout of a flat-headed fish. So I knew we found what we were looking for when I saw that. That was a good day. There are good days, there are bad days. That was a good day. Let's see, somebody's long. I don't want to, can't. For the discovery from Jason Sheppard, was only one viral gene repurposed for memory? I believe so. Yes, he talks about the one of them. You could do a Google search on ARC and Jason. You can see the original paper. It came out about 3 years ago. How could it be possible to determine if COVID-19 occurred naturally as opposed to experimentation in a biolab? Well, everything. Look, people have been talking about spillover from particularly viruses like coronavirus, you know, for years. You know, I mean, Laurie Garrett wrote a book in the mid-1990s called The Coming Plague, where she was focused on, you know, viral escape from wet market kind of things to society. David Quammen wrote a book called Spillover, which is exactly this, you know, a number of years, about 7 years ago. So, and if you look at the molecular sequence of the virus, it's similar to a bat virus. And so, I mean, Occam's razor, the simplest conclusion is that it likely came naturally from natural mutation from wet market, from bat, maybe some other vector in between. But, you know, can you absolutely exclude that it was, you know, didn't get released accidentally from a lab? Now you can't really exclude it, but I mean, does it seem likely? Probably not, given that all the other factors, it conforms exactly to what people have been predicting for years. So unfortunately, let's see. What are you most excited about exploring next? That's obviously the origin of vertebrates is a big one. One thing we're doing in the lab now is we're looking at regeneration and how regeneration can evolve. So it turns out that, salamanders, if you take a salamander, you cut off its limb, it'll regenerate pretty thoroughly. The muscles, the nerves, the bones, in high fidelity to the original pattern. Turns out fish can do that with their fins as well. And lots of other structures can regenerate in fish and things. So we're looking at the comparative biology and evolution of regeneration in the lab. That's what we were doing before things shut down about a month ago. And likewise, the origin of vertebrates in the field, that would be the next thing. Yeah, a lot of questions on viruses. I've been doing a lot of those. All right, well, I think. So what's on the horizon for life forms today? Do you have some bets on how today's structures will be repurposed for tomorrow? No, I wish I knew. One thing I can say, though, when you think about humans, natural selection is definitely acting on our genome. It's acting on the physiological traits, our relationship with microbes, as I said before. But, if we were all to get into a time machine and then, come back in 10,000 years, 50,000 years, and we'd see humans, what's driving the performance among humans? What's driving the differences among humans? How long we live, our cognitive capacity, our physical attributes, and so forth. It's hard to escape the fact that we have this, right? And we have inventions, we have cultural practices, we have educational practices, we have medicines, we have devices and tools that affect our performance. So much of our lives today are driven by human inventions and technologies. So when you think about it, human biology and culture and the ways we spread that information, to other people, that's sort of like an important factor. So like the yin and yang of human evolution is really our biology, so Darwinian evolution, but also the cultural practices, the stuff that results from the fruits of our brains, let's put it that way, our inventions and practices and devices and so forth. So I think future is largely under our control with our brains. So let's do right by it, huh?
I think we have time for about one more question.
All right, I am fishing around here. Let's see what we got. Have modern molecular tools revealed any concepts that Darwin got it wrong? Yeah, I mean, there's lots of areas. One of the things I should say right off the bat, do yourself a favor if you haven't, read Darwin's the 6th edition of The Origin of Species. It's an amazing piece of work. And in fact, it's read the 1st and 6th, but the 6th is kind of the definitive one. It's really amazing. One of the things that we see, the viral piece, he wrote a book before there was any theory of genetics, let alone DNA. Okay, and yet all his ideas apply to that, except for a few. That is, what we have is lateral, viruses can invade different species. So, the genes, so one of the proteins that's inside placentas came from a virus, as I told you, in ours. But it also invaded lizards as well, and they independently have that as well. There's no way Darwin could have known that, that the way that these genes can move from species to species via viruses. But what's even more remarkable than that, though, is how right he is, how many of his ideas that he came up with, in 1859 and before, apply so well to DNA. And he wrote it at a time that he didn't know even genes nor DNA existed. It's pretty amazing stuff.
Now I am not muted, and I'll say that again. Do you have any closing words before I take us out here?
No, I just want to thank everybody for coming tonight, and just wish you and your families and your loved ones the best at this time. And I look forward to a day when we can all be together.
Me too. So on behalf of the bookstore, the Harvard Division of Science, and the Cabot Science Library, I just really want to thank Neil tremendously, especially because it's his first book talk for Some Assembly Required, which is so exciting. And thank you to all of us for spending your evening with us tonight. We really do appreciate your support now and always. I also want to thank technology for working tonight. I'm really excited about it. Please make sure to check out Some Assembly Required at that green link below. And thank you again for your time and your purchases. So have a great night, everyone. Please stay well.
Thank you.